Yeah.
Good.
Okay. So, good afternoon. Sorry for all the technical issues. We're sorting through adding various machines to this infrastructure up here. Sometimes it works well and sometimes it doesn't. So we had a bit of a struggle this morning, but I think we're all set up now on Blackboard. I'm Fred Pryor. I'm the course director for this course. And I'm going to go through a couple of the quick housekeeping chores. First of all, for those of you who are connecting for continuing medical education credit, you need to get out of this ridiculous screen there. You need to call this phone number, 501-553-9108, and the event ID is 231202. So if you will do that, then you can connect to this course for CME credit. I also, because of the CME credit, have to discuss financial disclosures, and hopefully you can see this. We have no financial disclosures for Dr. June, who's our speaker, but I and Dr. Powell have financial disclosures as we were part of the organizing committee for the course, and so they are listed here. So having done all that background, I now, because of time, like to introduce our speaker, Dr. Saran June, who's a member of the Faculty of Biomedical Informatics. And she's going to speak on comparative genomic applications to metagenome inference and epidemic viruses. And I am going to turn the podium over to Dr. June.
Thank you. So, in this lecture, I'm going to talk about comparative genomics, focusing on applications. The number of genome sequenced each year is increasing exponentially. I believe that this is the good time to talk about where and how genome sequences can be used to attack what kind of problems. I'm going to talk about 3 different topics. One is Zika, Ebola virus. The other one is human microbiome. Okay. Let me start with Zika virus. What can you learn about the Zika virus from genome sequence? Zika virus is a positive sense single-stranded RNA virus, which belongs to the family Flaviviridae, the genus Flavivirus. And then Zika virus has its own name from Zika forest in Uganda. Zika virus was first identified in 1947 in the Zika forest, Uganda, and then it was found in mosquitoes and humans. Zika virus is primarily transmitted by the mosquitoes of the species Aedes. Aedes is also responsible for the spread of dengue and chikungunya. Recently, health officials in Texas have confirmed the first case of potential sexual transmission of Zika virus. Zika virus has been found in many regions in Pacific Islands, Africa, Southeast Asia, and Brazil. The map shows the distribution of Zika virus based on serology test and virus detection and laboratory diagnostic cases. The blue arrow represents a spread pattern of Zika virus, which was deduced from phylogenetic studies. We can use clinical symptoms to identify Zika virus. Many people infected with Zika virus are asymptomatic, or they have mild symptoms like fever, rash, joint pain, red eyes, muscle pain, or headaches. However, these clinical features are very similar to those of dengue and chikungunya. Therefore, clinical symptom-only based surveillance is not accurate at all. Recently, association of Zika virus with congenital abnormalities like microcephaly has been reported. Another test to identify Zika virus is a serology test. Serology test detect antibodies against the Zika virus. This test basically looks for the human body's reaction. However, this test, serology test, shows cross-reactivity between Zika and other flavivirus, like including dengue, yellow fever, St. Louis encephalitis, Japanese encephalitis, West Nile. The other laboratory test to identify Zika virus is PCR test. PCR test generates a signal for the presence or absence of viral RNA in a sample. This table I captured from FDA website. This table, basically they checked whether PCR test shows cross-reactivity. So this table cross-reactivity, and then this table shows the result says that not detected, which means that PCR test does not show cross-reactivity between Zika and other filavirus. This is another table I captured from FDA website. This is just part of the long table. This table, they checked this time again whether PCR test shows in silico cross-reactivity. So what they did is parse our sequences from forward primer and reverse primer and probe, and they did sequence comparison against of other flavivirus, dengue, yellow fever, West Nile, chikungunya, and so on. The number in the table represents the sequence identity of the best alignment. As you can see, the combined sequence identity is not high enough for homology, which means that PCR test does not show in silico cross-reactivity. We have been observing co-circulation of Zika and dengue and chikungunya, which means that where Zika outbreak is occurring is the same place where dengue or chikungunya outbreak is occurring. The figure, the left figure on top represent outbreaks up to 2005, and then the one right next to one represent outbreaks between 2005 and 2015. When we magnify region around Cook Island, we can see the overlapping spread pattern between Zika and chikungunya. So far, I presented a known fact about Zika virus. This is the first question we can ask. Do Zika genome show cross-reactivity with other flavivirus and chikungunya? To answer this question, we download all flavivirus plus chikungunya, which is the species in alpha virus, complete genomes, complete CDS available from NCBI. We obtained about 3,300 genomes for dengue, 162 chikungunya, 76 for Japanese encephalitis, 2 for KEDOC, and 6 for St. Louis encephalitis, 818 for West Nile, 36 for yellow fever, 80 for Zika, 1 for Spondwelli. So in total, we had about 4,500 genome sequences. To compare them all together, we first put everything into one frame, which is called multiple sequence alignment, denoted by X. And then we build the phylogenetic tree. To do that, we need to calculate the probability or likelihood of our observation, which is multiple sequence alignment, given a substitution model with the parameters theta and phylogenetic tree tau. Under the assumption that sites in multiple sequence alignment are independent of each other in terms of their evolution, the likelihood or probability is represented as the multiplication of likelihood of sites given substitution model and phylogenetic tree. We identify the tree which maximizes this likelihood. And then we explored many different evolutionary models like JC69, K80, and so on. And then use the information criteria to decide the best tree with balancing maximum likelihood and the number of parameters. First, we quickly check the tree of dengue genomes. As you can see, dengue, this tree showed distinction between different dengue types. We have so many dengue genomes in our data set, so we selected 2 genomes for each country for all species except for Zika. For Zika, we included all Zika genomes. Here's a maximum likelihood tree of filavirus and chikungunya virus genomes. So you can see that there's a clear distinction between species. So Zika genome does not mix with dengue or yellow fever or chikungunya. Here's our conclusion. The Zika virus has shown antibody cross-activity and similar clinical features with dengue and chikungunya, but genome sequences does not cross-talk. Therefore, the genome sequence can be used as a complementary tool for Zika virus identification. Considering the rapid geographic transmission, rapid geographic expansion of Zika virus transmission, and association of Zika with congenital abnormalities, we need novel sequencing technology for rapid detection and characterization of this RNA virus. So our group is working on third-generation sequencing technology for rapid pathogen identification from clinical samples. The second question we can ask is how Zika virus genomes are related. To answer this question, we need to root the tree. Generally, people use Spond-Wendy virus because Spond-Wendy and Zika belong to the same group, but Spond-Wendy is still not Zika virus. So people use Spond-Wendy virus root the tree after building a tree. But we did root-tip regression analysis under assumption of strict molecular clock. Strict molecular clock means that the genetic variation is strictly proportional to the time of the species. So this is the result of the best fit. You can, as you can see, the time of the species shows almost linear relationship with the genetic variation in Zika virus genome. The correlation score is 0.95. We use, so based on this analysis, we identified the root among the Zika genome to root the tree. So here's the rooted maximum likelihood tree of Zika virus genome. First, we observed the major two clades. One is for Africa, the other one is for Asia, Africa, Pacific Islands. We had isolated from Brazil, USA, China, Pacific Islands, Italy from the second lineage, but they are all mixed up in that lineage. The second observation we can make is about the isolates from Italy. We had 3 isolates in our data set. 3 of them grouped with Brazil, but 2 isolates which corresponded to 2 travelers. They traveled to Dominican Republic and then returned back to Italy. And 1 isolate corresponded to 1 traveler who also returned back from Brazil. So genome sequence can capture travel information. We had 2 USA isolates in our data set. So 1 USA isolate grouped with a clade of Brazil, Haiti, China. But actually this isolate was imported from Haiti. So it's expected situation. And the other USA isolate grouped with a clade of Mexico, Guatemala, Honduras. But this one also was imported from Guatemala. So again, genome sequence can capture travel information. Here's our conclusion. Genome sequences allow us to find the Zika virus epidemiological evidences. For example, we can trace the origin and spread pattern of Zika virus and potential spread of infection and travelers returning from the countries where Ebola outbreak occurred. So here's a cartoon of Zika virus genomes. Zika virus is a positive sense single-stranded RNA molecule of about 11K bases formed. There are 2 non-coding regions called 5' NCR, 3' NCR. Then the open reading frame of the Zika virus codes for 1 big polyprotein that is cleaved into capsid, precursor membrane, and envelope and non-structural proteins. Envelope protein E composes the majority of the virion surface involved with replication cases. So our question was, which genetic reason might be indicative of molecular evolution of Zika virus genome? To answer this question, we built a phylogenetic tree for each gene and compared the phylogenetic tree, and then we compared that with the whole genome tree. The heat map represents how similar genetic features are based on their evolution. We observed that NS5, non-structural protein NS5, shows the best similarity to whole genome in terms of their evolution. Okay, first question we can ask is, which genetic reasons are under positive selection? The comparative genomics is a powerful tool when we investigate the relationship between genotype and phenotype. In the case of protein coding genes, we can identify mutations that are fixed by population to change the phenotype. For example, enzyme activity or antigenicity of viral proteins. However, some mutations can be fixed just due to genetic drift or hitchhiking. So it is challenging to distinguish positive selection from natural selection. To identify sites under positive selection, this is the basic strategy. We formulate a null model first, which describes evolution in the absence of selection for some phenotypic change. And then we compare homologous sequences and then identify sites that deviate from the null model. So mutations in those selected sites might be selected because they need to change some phenotype. However, I believe that null model formulated by experimentally measured amino acid preference improves accuracy for positive selection greatly. But we do not have null model based on experimental measure, so we formulate null model this way. To answer which genomic reason might be under positive selection, here's the null model we set up. Sites can be classified into two categories. Sites are under natural selection or they under neutral selection. Omega is the ratio of synonymous mutation rate to synonymous mutation rate. Naturally, alternative model describes that sites can be under either natural selection or neutral selection or positive selection. To make sure whether genes of interest contain the sites under positive selection, we did likelihood duration test. This whole process is called site-specific codon model. We performed site-specific codon model at the genome scale. But here's the output. We did perform site-specific codon model, but we did not find a single gene which passed the likelihood duration test with a p-value which is less than 0.01. But still we can make a list of sites whose posterior probability of omega larger than 1 is at least 0.5. So this is a site under positive selection, but this doesn't have strong signal to make sure whether this selection is not random. But I still make a list of sites under positive selection. We identified one site in C, capsid gene, and one site in NS1, 3 sites in NS3, and so on. Okay, fifth question we can ask is, which genomic regions are under positive selection along the specific branches? Let's say we are interested in the lineage, which contains isolated from Asia, Americas, Pacific Islands. We want to identify sites under positive selection, focusing on their lineage. To answer this question, we set up two models, alternative, null model. Alternative model alternative model allows sites to be under positive selection for a class of codons for focusing on that specific branch. null model does not allow such a selection. This kind of procedure is called branch-side codon model. We performed this branch-side codon model at the genome scale, and then we did a likelihood ratio test to make sure whether the selected site is selected by random or not. But we did not find any single gene which passed likelihood ratio test with the p-value less than 0.01. But this is a list of sites whose posterior probability is at least 0.5. We identified one site, NS5, whose posterior probability is about 0.9. Okay, there's no medicine for Zika virus infection. So epitope is a part of antigen recognized by the immune system, specifically by B cell and T cell. We are working on B cell and T cell epitope prediction using sequence-based methods, including machine learning, artificial neural network. This output prediction might be helpful for the development of a peptide vaccine. But I will describe T cell epitope prediction in Ebola virus soon. Okay, I'll move to the Ebola virus. What can you learn about the Ebola virus from genome sequence? Ebola virus belongs to the family Bullovidae, and the genus Ebola virus, species Gyre Ebola virus. When I say Ebola virus, it means Gyre Ebola virus. Gyre Ebola virus is responsible for the 2014 Ebola outbreak in West Africa, which was the largest documented one for Ebola outbreak. Like we did with Zika virus, we first compared more than 100 Gyre Ebola virus genomes to other all viral genomes with the family Bullovidae. The genera in that family, Marburg virus, Ebola virus, Quebo virus, forms distinct groups, and then species in the Ebola virus. There are 5 species in the Ebola virus: Western Sudan, Gyre, Tigris, Bundibogo. They all form the distinct groups, like the Zika virus. And then we move to the Gyre Ebola virus, and then check the relationship between Gyre Ebola virus. This is the rooted tree of the Gyre Ebola virus genome. This tree is rooted to the earliest recorded one, because that one shows the best relationship between the genetic distance from the root and the time of the species. First, we observed 2 major lineages. One lineage is for Central Africa. The other one is for West Africa, which contains 2014 Ebola outbreak. And then we observed 5 lineages, 5 major clusters, which contains isolated from 5 distinct periods. And then we observed that 2014 Ebola outbreak forms a very distinct cluster instead of being nested within the previous Gyre Ebola virus. So that we speculate that 2014 Ebola outbreak caused by very divergent means of Gyre Ebola virus. Here's a cartoon of Ebola virus genomes. Again, we asked which genomic region might be indicative of a molecular evolution of Ebola virus genome. To answer the question, we considered many different genomic regions, coding genes, proteins, intergenic regions, and then build a tree for each genomic feature and compare that to the whole genome tree. So as you can see, the white area corresponding to intergenic regions. So all intergenic regions shows very different tree topology from trees of coding genes and proteins. And then glycoprotein coding gene, that gene shows the very similar tree topology of whole genome tree. So we speculate that the GP, glycoprotein gene, might be indicative of molecular evolution of giant Ebola virus. We performed the T-cell epitope prediction. There are two kinds of immune responses. One is humoral immune response. The other one is cellular immune response. Humoral response is mediated by the B cell, which eliminates extracellular pathogens, and cellular immune response is mediated by T cell, which kills intracellular pathogens. And then we did the T cell epitope prediction. So I'll talk about only T cell epitope prediction. T cell bind to a particular antigen displayed on the surface of infected cells in conjunction with major histocompatibility complex, MHC molecules. In human, MHC molecule is called HLA, human leukocyte antigen. There are two types of T cytotoxic T cell and helper T cell. And then there are two types of MHC molecules, MHC class 1 and MHC class 2. So cytotoxic T cell binds to a peptide in conjunction with MHC class 1 molecule, and helper T cell binds to an epitope in conjunction with MHC class 2 molecule. The only AIMer or NIMer peptide can fit peptide binding group of NHC class 1, and 12 to 17 mer peptides can fit peptide binding group of NHC class 2 molecule. So peptide binding to NHC is the most selective step for the peptide to become epitope that induce a T-cell mediated immune response. So we are trying to identify peptide in Ebola virus genome, which fit a peptide binding growth of NHC molecules. That is called the T-cell epitope prediction. So in the first step, we collected HLA alleles specific for the US and West Africa population, along with allele frequency data from the allele frequency net database. However, MHC molecular variance in human population is huge. That can cause different human responses. So we can factor allele frequency data for Caucasians, Hispanics, African Americans, Asians, and Native Americans. We focused on only NIMer peptides for NHC class 1, and we focused on only 50 mer peptides for NHC class 2. And we used the method developed by our collaborators in Denmark to predict the T-cell epitope in Ebola virus genome. We used two thresholds, weak and strong threshold, because strong threshold sometimes can filter out virus inactivating epitopes. So we applied two thresholds to select predict epitopes. In third step, we calculated the epitope score for each amino acid in Ebola virus protein. The epitope score is defined as the number of predicted epitopes whose positions overlap with the given amino acid position. And then we normalize this score by allele frequency to give epitope bound by prevalent NHC a larger contribution to the score. So we made epitope selection for each population. So we identified epitopes specific to the US and West Africa population. We also identified epitopes which are strongly conserved and recognized in both populations. And we did a research survey to make sure, to check whether each epitope were experimentally validated. So our prediction picked up 3 MHC class 1 epitopes from GP, glycoprotein, which were experimentally validated in mice. But we also our method used one epitope derived from nuclear protein. Okay, we picked up predicted epitopes based on their population coverage to map them onto Ebola virus genome. The red means predicted NACC class 1 epitopes from US and West Africa. Blue means predicted NACC class 2 epitopes from US and West Africa. We identified the 7 epitopes in the L protein, and none was detected in BP24 for NACC class 1. Interestingly, we identified the regions in MP and L that were recognized as both predicted NAC class 1 epitope and NAC class 2 epitope. Okay, I'll move to the human microbiome. Can we derive functional structure of human microbiome from 6' amplicon sequencing? The human microbiome is composed of more than 100 trillion microorganisms, which colonize our skin, gut, mouth, elsewhere in our body. It is a fact that the microbiome is important for maintaining human health, and it can contribute to disease when things go wrong. How can you study the microbial community? Rapidly developing technologies are enhancing our ability to study microbial community. There are 2 ways to explore the microbial community. One is shotgun metagenomic sequencing. The other one is 6' ribosomal RNA amplicon sequencing. Shotgun metagenomic sequencing a complete list of all genes in all organisms present in a microbial community. That method can answer which species, which functions are present or absent. However, enough metagenomic sequencing to capture reasonable amount of DNA sequences carried by every single organism present in an organism is very expensive. So the other method to explore microbial community is 6' ribosomal RNA amplicon sequencing. This method is very rapid and cheap. This method is about 1,000 times cheaper than shotgun metagenomic sequencing. This method can answer bacterial diversity with their abundance. However, we cannot derive functional or metabolic capabilities directly from this one gene study. So we developed a method called PanFP to infer functional profile of microbial community from 6' amplicon sequencing. Here is our output table from 6' ribosomal RNA amplicon sequencing. This table is called OTU table or 6' ribosomal RNA survey data. Let's say we have 10 samples from skin, S1, S2, S10. And OTU means operational taxonomy unit. The number in a table means occurrence in a given sample. Each OTU has a taxonomy information. The motivation behind our method is taxonomy information can capture clusters due to the shared function content. In the first step, we first generate a database of genomes for pan-genome construction. What is a pan-genome? Pan-genome is a set of all genes present in a given set of organisms. So we downloaded all bacteria, archaea, chromosome proteins available from GenBank, and we used the KAG authors database for genome functional annotation. When you go to KAG database organize functional terms hierarchically. There are 6 categories at the top level: metabolism, cellular processes, and so on. When you click one category, you can see the lower level of categories. And when you click for example, cancer, you can see a list of pathways for that category cancer. Let's say when you click the one pathway, then you can see a list of k-terms which are involved in that pathway. We use these k-terms to annotate our genome. So we annotated the genome using k-terms, and then we keep only with genomes with at least 30% of functional coverage, which is defined as a fraction of proteins with a functional annotation. Okay, so we generate a database of genomes for genome construction, and then we modify the taxonomy information by abandoning abandoning taxonomy letters from the bottom until we can identify at least one genome in our database which belong to the given taxonomy. So, for example, we don't have a genome in our database which belongs to the Salina archaea. For that case, we drop the genus information. So we modify the taxonomy information. And then we build a pan-genome for each OTU by collecting genomes from the database which belong to the given taxonomy. And then build a pan-genome function profile for each OTU. The frequency of kiloton in pan-genome is defined as occurrence of kiloton in pan-genome, and then divided by the number of genomes pulled for pan-genome construction. So the kilotons more shared by genomes pulled for pan-genome construction will have a larger frequency. Okay, we build a pan-genome for each OTU, and we build a pan-genome function profile for each OTU. And then we did normalization, because the number of the sixness type number varies a lot from 1 to 15. So we normalized the raw frequency of OTU by the putative sixness ribosomal RNA copy number, and then normalized again the number by sample size. So the final number represents organismal abundance, which are comparable across all different samples. In the last step, we combine pan-genome function profiles with weights which are corresponding to the normalized abundance. To validate our method, we collected 65 microbial communities, 39 samples from mammal gut, 14 from soil, 10 from hypersaline microbial nets, and 2 from HMP mark communities. For each sample, we inferred functional profile from sixness amplicon sequencing, and then compare that with the functional profile obtained from direct shotgun metagenomic measurement. For HMP, the correlation score reaches about 0.9. I included this page just to show our inferred function profile captures geographical location and environmental characteristics. We applied our method to about 500 populatory samples. We collected this sample from 2 different geographical locations and 3 different environments, and then applied our method and infer functional profile for each sample. And then we did the PCA analysis. And these are PCA plots. As you can see, the samples form the 6 different distinct clusters, which means that our input function profile captures geographic location and environmental characteristics. Here's our conclusion. Our method allows to perform comparative inferred metagenome analysis. We can highlight statistically which functions are the most overrepresented and which functions show the big difference across different samples. from sequence viral RNA sequencing. And we can use our method to serve as a first step to identify potential samples of interest for large-scale metagenomic projects. Okay, here are 5 quizzes. You can find the answers on the blackboard. I hope I presented at least a little of flavor of importance and overall application of comparative genomics. Thank you.
Any questions for our speaker? Yeah, so I may not have understood, so I missed it. I apologize. So with cross-react, so if I missed it, I apologize. But with the cross-reactivity, you did the phylogenomic analysis, and then I wasn't understanding how you were connecting, creating the phylogenetic tree cross-reactivity.
So we built a phylogenetic tree of Zika viral genomes. We built a phylogenetic tree of filavirus genomes, including chikungunya, which is a species in alpha virus. So we included the filavirus and chikungunya virus in the analysis because clinical features and serological tests shows cross-activity between Zika and dengue and chikungunya. So we included all filavirus genomes and chikungunya genome and build the phylogenetic tree, and we observed that all species in our data set formed distinct groups, meaning that, which means that, let's say you give me a sequence, then I can tell, or I can tell which species this sequence belongs to with high accuracy.
So my understanding of cross-reactivity is that the immune system can't tell the difference between, say, this virus and that virus. So the same systems are being elicited from the infection. So how does there being very distinct groups connect to the immune system's inability to distinguish those groups?
So, okay, maybe, okay, I need to say it this way. So, genome sequence do not crosstalk. That's what I mean by cross reactivity. So, if you give me a genome sequence, then I can tell whether this one is Zika or dengue or chikungunya.
I see.
Yeah. Okay.
I misunderstood what was meant by cross-reactivity.
Okay.
I have a question about the microbiome
Yes.
study with it. You had 39 mammalian gut samples.
Yes.
What type of mammals, and what's the clinical significance of the study?
So okay. You mean this one, right? Okay. So we cannot derive functional and metabolic capabilities directly from 6-ness amplified sequencing because this is one gene study. But we want to, but this method can infer functional profile for a microbial community from 6-ness amplified sequencing. So I need to validate our method. I used these samples to validate our method. So I do not remember the detail about this 39 mammal samples, but this sample has two informations, like 6-ness sequencing and metagenomic sequencing, which means that I know the function profile obtained from the shotgun metagenomic sequencing, and then I have OTU profile from 6-nest sequencing. And then I infer function profile from 6-nest sequencing and then compare that to function profile obtained from management sequencing. But the correlation score for these samples is not that high compared to the one for HNP because the sequencing depth, management sequencing depth for this sample is not high enough, meaning that not enough DNA sequences for this sample. So it is expected that it is expected not to have high correlation scores. Yes.
So coming back to Zika, it looks like someplace in this transition from Asia to, well, maybe from Africa to Asia, from Asia to South America, it evolved from being purely mosquito-borne
Yes.
to a sexually transmitted disease. Is there any indication of a genomic change that might explain that?
Actually, that's the one we are working on right now. So we want to identify any genetic evidence for sexual transmission. So I don't have an answer for that, but we are investigating that information-based genomes.
Any other questions? The audience outside the room? Well, thank you all very much for joining us today, and thank our speaker again.
Thank you.